introduction for getting started with CellProfiler. CellProfiler is a tool that was made by biologists together with computer scientists, probably originally made for analyzing cells in 2D culture under the microscope, but can be used for all sorts of other things. There's a whole worm toolbox for nematodes, and the principles of image analysis that it uses can be applied to all sorts of different things. I'm going to talk about cells, two-dimensional cells in culture with a nucleus and a cytoplasm and how we can find them. But again, this is just going to be one use case for CellProfiler. No, it says cell in the name, but you could look at other things as well. And this is only going to be kind of a snapshot of what CellProfiler can do, but hopefully enough to get people started if they are interested. So let me minimize myself here. And the very first thing to start with is how do I get CellProfiler? So if you go to cellprofiler.org, you can download CellProfiler for free. Mac and Windows. If you want to install it on a university computer, you'll probably have to talk to IT about that, raise an IT ticket, but you can download this on a laptop, a home PC, and there's quite good documentation. There's getting started, there are tutorials, there are example data sets, there are manuals, and there is support. So there's a forum, which is pretty actively used. And even you might get questions answered by the developers themselves. So this can be found at the ImageSC forum, and there's a whole section on CellProfiler. And quite often it is actually like the designers and people who maintain and update. So Beth Simony, for example, is one of the head hunters of CellProfiler. So you really get quite a lot of support as well as really good documentation here. Okay, so once you've downloaded, you'll get the program. I have just opened a very fresh CellProfiler. So you'll get on some computers, this does it on my Windows computer. I don't think it pops up on my Mac. Don't close this little terminal, just minimize it. When you first open it, you'll get a pop-up. You can say, don't show this again, but it has even more tutorials, examples, getting started, Q&A, manual. So even if you're not looking on the website, you've got a lot of this information available to you. But what we want to do here, I want to take you through setting up some new data and then finding things, nuclei and cells in particular, in that data. So CellProfiler is made, it's open source. It can interact with lots of different files. It can mix and match 8-bit images, 16-bit images, RGB images, and combine those into whole image sets that you can mix and match and analyze. This is not always the case. Like in ImageJ, ImageJ does not like to combine 16-bit and 8-bit images. CellProfiler can do a lot. There's one hurdle though, and it's at the very, very beginning, which is that you have to tell it what it's looking at. If you're using, say, the Zen software and you're doing some image analysis in that, you don't have to tell it anything because the file contains all the metadata. So you don't have to tell it that there's 10 sets of images and each of those images has 3 fluorescence channels because it already knows. But CellProfiler doesn't know. So first thing we're going to do is we're going to put some data in there. So here I've got, in a demo, I have got 2 sets with 3 fluorescence channels each. So they've got this H3342, that's Herx 3342. There's a Psi3 channel, that's a red channel, that's got an actin stain. And here the AF647, that's Alexaflor 647. And that is, in this data set, labeling cells which are proliferating, so cells which have incorporated EDU. but it could be anything. So the first thing we're going to do is just drag and drop to load the files. Now, confusion that often happens. We drag and drop, we put it in the box, but we haven't put the files into the CellProfiler box. That drag and drop is just a graphical user interface tool to tell CellProfiler where our files are without having to type them out. So if you look here at this little line, which I feel like as biologists we tend to ignore, but this is actually saying this is where it's looking. It's looking in this particular drive in this particular folder. So if I sent you a pipeline that I'd written with images loaded into it, you would then have to, once you downloaded those things, remove the files from here, so we can highlight them, right-click, remove them from file list, and then you would have to bring them in from wherever they were stored on your computer, because presumably your folders would not be called C users JS348 documents CellProfiler nuclei demo. So you have to tell CellProfiler where to look. You can also double click on the images and see what they are. There are some tools here which can be useful. So there's a magnifying glass, you can zoom in. You can change how big the image is, how much room it takes up on the pop-up. You can right-click and adjust the brightness and the contrast. If maybe something is not very visible, you can make it brighter. This is also quite helpful because you can see kind of how much of the full dynamic range, with where 0 is black and 55 is white. You can see how much of the dynamic range that image is taking up. You can also use this little measuring tool to, say, measure the size of the nuclei, which I'm seeing here. So if you look down at the bottom of this box, you can see there's an X position, a Y position, an intensity, and a length. So that length is 108. Its length is 50. So this is very useful because when we start to ask CellProfiler to identify objects, we are going to need to tell it how big, roughly, are the things that it's looking for. Are they a few pixels wide? Are they 1,000 pixels wide? So this is giving us the length in pixels. We might look at another image. And see that this is a bit dimmer, but we can see a nice bright actin stain there. Okay, so once we've done that, we've got the images in. Now we need to extract some information, usually from the file name, although you can also extract information from the folder name. Now, if all I had here were, single fields of view with a DNA stain, and all I wanted to do was count the cells, I might skip this step because all of the files are the same. However, when I exported that data, it's just going to say image 1, image 2, image 3, image 4, image 5, etc. It's not going to say I have a 96-well plate. It's not going to tell me what well it came from. So I'm going to say extract metadata. And I'm going to write a little command that's going to tell the software how to read these file names. So there's some important information in these file names. This is taken from a 384-well plate. So we've got some really important information. So J19 and L19 here refer to the well, where the letters are a row and the numbers are a column. We've also got for J19, we've got 3 fluorescent channels. You can also see there's a P1 and a P3 here. So within a single well, so within L19, this is position 1. Within well J19, this is position 3 out of maybe 9 positions, however many positions in the well that we took. Here we also have a scene. This is the way that the Zeiss Zen software organizes things. It tells us basically the order in which that image was taken. So this was a lot of images in a 3D four-well plate. This is the 638th set of images, the 638th position. This is the 806th position. So the first thing we want to do is we want to tell CellProfiler how to get that information out. And we're going to use a tool called regular expression, or Regex. And Regex is great because it actually works in a lot of different software. So you can use Regex in ImageJ. You can use Regex all over the place. It's a common way. So I like to use this Regex 101 in order to write my Regex. So it's a bit of a learning curve, but once you got the hang of it, it's kind of fun. It's like a fun little game. So we are going to write this regular expression and pull out different pieces of information. So to start the regular expression, we need to use this little up arrow. And you can see that there's an explanation over here. So there's a lot of information. So this says, asserts the position at the start of a line. So this is like, we're beginning a command here. So now there's some letters and numbers that are common to, save that one for later, that are common to all of the file names. So they all say demo 2025-scene-. So we can just say, yep, scan along, read demo 2025-scene-. Okay, so it's getting highlighted here in blue. So this is say match a string, test string. In this context, a string is characters, so letters, numbers, special characters that are not a command. So we're just saying, yep, scan along, read all of this, the text that literally matches these letters and numbers. And now we're going to make our first capture group because we want to pull out the information about what number scene this is. So a capture group is just saying we're going to capture some information and make a group out of it. So to do that, we're going to start with brackets. To start a new capture group is $P. I don't know why, it just is. And so we're going to need 3 pieces of information for the capture group. First, we're going to give it a name. You could call it anything you want. You could call it Fred, you could call it Jeff, but I'm going to call it scene because that is a name that makes sense. And we're going to do that in these greater than, less than, or pointy arrow brackets. Next, we want to tell the regex what kind of characters we're looking for. And here, if you look down on the quick reference, there's all sorts of different options. So what we're going to look for here are numerals. So this information goes in square brackets. So I'm going to say we're looking for numerals 0 to 9. And then the final piece of information we need to give it is how many numerals or how many characters are we looking for. And here we are looking for 4 characters, and we're going to put that in curly brackets. And now when I finish the brackets, we should have a new capture group. Yes, we do, and it's called scene, and the value is 0638. Okay, so now we've got some more things which are common. We've got -p for position. And now we want to make a new capture group. So that's going to be $P. And we're going to call that position. And look what I'm typing. And now position, again, we're looking for numerals, but we're just looking for one of them. So great, now we've got position 3. Okay, and then there's another dash that's common, so just as a string. And now we're going to pull out the row and the column. So capture group again for row. And for row, we are looking now not for numbers, but for letters. And specifically, we're looking for uppercase letters. So I'm going to say uppercase A to Z in square brackets, and we are looking for just one of those. Not exclamation point, but one. Okay, great. So now not skipping anything, just going straight into our column capture group. And here we are looking again for numerals. and we're looking for 2 of them. Okay, so we've got some more bits of string, some more characters which are common to all of our file names. So we've got image export, and then we've got a dash. For whatever reason, when the Zeiss software exports files, it's giving us both the scene, and it's also telling us the order in which it has exported this group of images, and so this was the 638th export. Also happens to be the 638th scene because it's exporting it in the order that it took the images. It's a little bit complicated here. In both of the image sets here that we have examples of, there are 3 digits. But I know from looking at the original files that it goes 0, 1, 0, 2, 0, 3, blah, 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 10, 11, 12, 99, 100. So we can either have in this little bit, we can either have 2 numerals, 3 numerals, or 4. Actually, no, yeah, there might even be 4 numerals. So I don't need to make this a new capture group though, because I've already captured the scene. So I don't really need this piece of information again, but I want the regex to sort of scan across it. It's going to be different for each file name. So I'm just going to put a brackets, but not a question mark P. So I'm not making a capture group. And I'm going to say, okay, just scan along, look for some numerals. And I could put 3 here. So this is just a group, but it's not a named group. It's not a named capture group. And this works fine. But if I was analyzing my whole set of data, it would break the regex if it saw group 38 or group 1038. Or if it was group 1038, it would catch 103, and then it would break downstream of that. So what we can do here is instead we'll tell it anywhere from 2 as many as 4 numerals. It's not 2-4, it's 2, 4. So it's 2 numerals or 4 numerals, whichever comes second before you get to the next thing. Next thing is an underscore, and then we have the channel. Now the channel is going to get also a little bit complicated because if you look at the file names here, we've got H3342, we've got AF647, we've got capital C, lowercase y, 3. So we've got a mixture of uppercase and lowercase. We've got a mixture of letters and numerals. And we have a couple of different options here. We could say, give me A to Z, or A to Z, 0 to 9, and give me 5 of them. So it finds that. Totally legitimate way of doing it. We know that from our file names, we've got Sci3 and we've got H3342 and AF647. So we want to tell it, look for 3 to 5. And now if we change this to Sci3, hopefully we still get Sci3 as the named capture group there. If you don't feel like writing out all of these different possibilities, you can say any non-white space character. So a letter, a number, uppercase, lowercase, a special character, anything, as long as it's not just a space. And that is denoted by, let's see, slash capital S. So that, oh, well, that kind of worked, didn't it? That would have worked great for AF647, but see what happened here with Sci3. It's going to call this channel Sci3_O, which is fine. I mean, if you don't mind it doing that, then that's fine. But if you do mind that, then you can be more specific. And it will give you exactly what you're looking for. There is some more bits of text, bits of string at the end of this, but don't care because we don't really need them, so we can stop here. So what I'm going to do is I'm going to copy this regular expression and go back into CellProfiler and go to the Metadata tab and put that in here. And now when I click Update, we've got some information. So here, every row is a file. And every row now has a column, a row, a scene, a well, a position within the well, and a channel. Great. So we've got all of the information that we need out of the file names. Now we need to tell CellProfiler how to group these, because it's not clever enough to just say, well, obviously, yeah, you're going to group by well and position. It doesn't know that. So we're going to need to tell it that in each set of images that correspond to one position, that there are 3 channels, and they correspond to each of these 3 channels. So if we go to names and types, we can say, assign names to images matching rules. And now this is going to let us label what our different channels are. So we can say metadata has channel matching. And here I'll say H3342. And this is my DNA image. So if I hit update, yep, we've got 2 DNA images. So we can say add another image to the set as metadata. Metadata has channel matching AF647. And here I'm going to call that the EDU because that's what it is. And now we've got a DNA and an EDU image. And if we were like, okay, actually, this data set has 3 channels, but we're really only going to use 2 of them, we could stop there and it'll just ignore the other one. But we do actually want to use all 3. So I'm going to add another image. And again, say it has metadata with the channel matching Psi3. And watch out because all of these are case sensitive. And that is my actin stain. And so now we've got 2 sets of 3 images corresponding to the actin, the DNA, and the EDU. There's another bit called groups, which we're not going to use at this time because I want to get straight into actually using CellProfiler to find things in the images. So I'm just going to minimize this and go to a pipeline that I've already made. And here you can see that above this line, this is like the giving information to CellProfiler, but below this line is where I can add modules. And actually, we'll just go into this one so you can see how you actually do that. So we can go to, it's called adjust modules for some reason rather than add modules, but if we go and we hover our mouse over, we can still go to add module. And now we can do some all of our actions that we want to do. So we might want to first say, all right, we want to identify our primary objects. And when we double click on something, it adds it to the pipeline. If you want to know what something is, you can just go to this module help, which actually very helpful. It gives you a lot of information on what that module does, what it is, and there's all sorts of things that you can change in that module. And it's going to give you a lot of information about that too. So this is really, really helpful. Also, see these question marks? You can always click on these question marks and they will give you some information. I typically go straight to the advanced settings, which looks a little bit intimidating, but it's not that bad. And the nice thing about this CellProfiler image analysis stuff is like you could just go through and change everything one at a time, go into the test mode, and just see what it does. You're not going to break anything. You can save as many versions of the pipeline as you want. They're not big files. You know, you can save test 1, test 2, test 3, whatever. It's a good idea, actually, if you're playing around with parameters to save different versions of those parameters. And, if you're going to be really on the ball, take some notes about what you changed and what effect that had that you can come back to later. Okay, so that's how we add things. So once we're here and we want to see what things do, we can go into the test mode. When we're in test mode, we've got some options. So we've got this little tick box, which is going to enable that particular module, which is going to run it. We've also got the little eyeball. So if the eyeball is open, when we step, it's going to show us what it did. So, oh, you know why? Because I unticked the box and then I stepped. Okay, so let's go here. Identify primary objects. Sometimes the pop-up box hides, especially if you have got a lot of other windows open. And then again. Hey, where is it? Where'd it go? Oh, here it is. Got minimized somehow. Okay, so here we can see this pop-up where we have our input image with the DNA stain. And here are the objects that it found. And we can zoom in if you want to see that a bit clearer. We can see the outline around the nuclei, and we can see each of these different colors, just a random color showing us the different objects. Okay, cool. So how did we get there? All right, so the very first thing that we need to do is tell it what the input image is. So here we've got a choice of DNA, actin, and EDU. So we'll say DNA. Now you can name the objects, so you might want to call them nuclei. I'm going to call them nuclei 1 because later, after I've found the nuclei in the cells, I'm going to do a filter so that when I make measurements, I'm not measuring any cell that gets cut off by being on the side of the frame. So I'm going to call that nuclei because that's going to be like my final nuclei that I'm making measurements on. So I'm going to call this here nuclei 1. And then we have probably the most important thing, which is the typical diameter of the objects, which we can measure again anytime we want to with this measuring tool. I've made it quite a broad range so that it catches very small, like metaphase and anaphase chromosomes, all the way up to some very large nuclei. I'm going to discard objects outside of this diameter range so that anything that's just like a little piece of maybe debris of a dead cell, or sometimes there's little fuzzies that get in that are much, much bigger than a cell. So it's going to discard things out of that range. I'm not going to discard objects touching the border at this stage because I'm going to do that after I find the cells, because I don't want to I don't want to confuse my cell finder by counting merging cells that are on the edge of the image. So I'm gonna keep everything for now. Threshold strategy. These defaults usually work pretty well, but there are things that you can play around with. And again, the question mark here is your friend. And also just pressing the buttons and changing things and seeing what happens is your friend. Other important things here are the smoothing scale. If you find that you're getting objects with very jaggedy edges, applying some smoothing is good. If you've got maybe nuclei or objects that have some bright spots and some dark spots, the smoothing also helps so that you're not getting kind of holes and little jaggedy edges or insertions in your object. The threshold correction factor, basically if this number is lower then it's going to be more lenient. So you can imagine the threshold, like the pixel intensity, as a hill. If you take a line across the nucleus, so if you drew a line, say like this, and then you plotted the pixel intensity, you would have something that looks like a hill or a top hat. And so the threshold is going to be how far up that hill or down that hill are you going to draw the line for being inside the nucleus or outside the nucleus. So if we make this number larger, it's going to give us smaller nuclei, and if we make the number smaller, it's going to give us larger nuclei. And again, if I've just said this wrong, you can always go to the question mark and it tells you. There's some other things that you can change. If you've got very weak signal, log transforming all of the pixel intensities before thresholding can help you amplify the difference between the foreground and the background. Method to distinguish clumped objects and draw dividing lines through them. You've got choice of intensity, shape, or none, so it'll kind of be neutral. I tend to use shape. It will automatically calculate the size of the filter for the declumping, which is like drawing lines between different objects. The default is usually pretty good, but it might not be for yours. Something else that's quite helpful is saying don't automatically calculate minimum allowed distance between local maximums. There's a fancy way of saying there's going to be some bright spots in your objects. Each object will have like a brightest spot, a next brightest spot. So these are the local maxima. Smoothing is going to kind of smooth out those peaks and troughs a bit, but there will still be local high points. And you can tell it here that you want objects to be a certain number of pixels apart. So if we make this number very small and we step through, takes a bit of time to run when I've also got the recording on, I'm sorry. So see what happened here. When I made this number very small, it's cutting objects in half. But if I make it larger, it's going to not cut objects in half. So if you see that maybe it's merging cells too much, then maybe that number is too big and you want to make it smaller. Okay, so once you're happy, it may never be 100%, but hopefully you'll get sort of 98%. And you might want to take that opportunity to save, or save a version where you've put these particular parameters in. And then we can move on to identifying the secondary objects. So primary objects, kind of as the name suggests, are bright blobs on a dark background usually. If you have, say, GFP positive neutrophils, you don't have a nuclear marker, but you've got pretty well-defined, bright green blobs on a dark background, then you can just go identify primary object, and your primary object is a cell. But here our primary object are nuclei, and then we're going to use these nuclei as seeds or kernels, which is the literal meaning of nucleus, and then to expand outward using a label that stains the cytoplasm. So in this case, that's going to be actin. So again, our first thing we do is select our input image. That's going to be actin. Select the input object. So that's the object that you'll use as a seed. So there you go. And that's nuclei 1. I'm going to call these cells 1 because, again, we're going to filter. So let's just step through. Usually secondary object takes a bit longer. It's more computationally intensive. So you might see the little blue pinwheel going for a bit longer, especially if you're running lots and lots of programs at the same time. So while that's doing what it's doing, we can talk about some of the parameters that you can change. So there's the smoothing scale. This is usually pretty important actually with cytoplasm. If it's it's 0, you'll probably end up with quite jagged edges, so you want to apply some sort of blur. Threshold correction factor, same as before. Log transform before thresholding, same as before. Regularization factor. This is something that you might want to play around with if your cells are touching each other a lot, and you can read a bit more about the regularization factor, change it, see what happens. You can ask it, do you want to fill in the holes, or are your objects things that have holes in it? And at this point, you can say, do you want to discard things that are touching the border of the image? I did not say yes here. I have a filter objects later, but you could do it here as well. So let's see what we got from secondary objects. So usually your first pass at secondary objects is going to be not good. And here we can see that this is not great. It's not terrible, but it could be a lot better. And there's a couple of reasons for this. So this is an actin stain. Actin is actually not the best thing to use for a cytoplasmic stain. Best thing to use for a cytoplasm stain is something that's quite uniform across the cytoplasm. And F-actin actually isn't, because here we've got quite bright F-actin around the nucleus, a bit of a dim patch, and then bright actin again around the periphery of the cell. Like this cell that has got a lot of stress fibers, a bit easier to see. This cell that's sort of uniformly bright, but you can see it's really losing the plot when it's trying to segment this particular cell here. So you could play around with these parameters, but another trick that we can use is to rescale the intensity. So I'm going to put this rescale intensity step here, and if you want to know where that is in image processing. You can also look it up, so rescale. You can also see all of the different modules that you could use, and you can get information about any of them. So I'm going to rescale this image because I don't really care how bright the actin is. I just want to make some sort of binary cutoff about what is foreground and what is background. So I'm going to look at my actin image here. And as I move the cursor around, if you look on the bottom, you can see the x coordinate, the y coordinate, and also the intensity. So the intensity of the background is about 0.2 plus or minus. Now, why is this a decimal place? So I said that CellProfiler is kind of format agnostic. So it can take an 8-bit image, a 14-bit image, a 16-bit image, a 32-bit image, and it can work with them all at the same time, even in the same image set. This is very helpful, but it's a little bit disconcerting if you're used to looking at intensities values which are integers, which is 139 out of 255, or 7,500 out of 65,000 for whatever, for a 16-bit image. So what CellProfiler does is it takes whatever the bit depth is, and it just rescales it from 0 to 1. And that means that it can work with any bit depth. But it also means that in a 16-bit image, that maybe the highest intensity is 12,000, you're going to have quite a lot of that number line being zeros. So you're going to end up with quite a small little decimal points. You can always change them back into integers if you want to express data in integer form or like do math on them. So here we can see that our background is about 0.2. Some bright actin is 10 times brighter than that, so 0.21, so that's pretty good signal to noise. But over in these dim parts of the cell, it's 0.3 compared to 0.2. So we can rescale the intensity in various ways. You can stretch each image to use the full intensity range. I tried that. It was not actually great because there were a couple just really bright spots. And so it was stretching it to the min and the max, but there were some really high max outliers from like little bits of dead cell debris. So there's other ways that you can do it. You can divide by the minimum, the maximum, match it to another image. I'm just going to choose specific values to reset to the full intensity range. So basically I'm going to say, okay, reset 0.15, call that 0, and 0.06, call that 1. So let's see what happens there. Okay, so you can see that the rescaled image, it shows up a lot brighter. And so what we want is just that our foreground is much, much brighter than our background. And so, we could play around with that. We could play around with the levels and just see what gives us a nice differential between the foreground and the background. So that looks pretty good to me, so let's use that. And so now I'm going to call that rescale intensity actin, and I'm going to go here and say, please use the rescaled actin image to find the secondary objects. Now, caveat on the rescaled images. Rescale things, do whatever you want to them to get the segmentation that you need in order to find the objects that you need. But when it comes time to do intensity measurements, make sure that you're measuring the intensity on the original, not the rescaled image, because right when you rescale it, you're losing information. Yeah, here's our new one. Okay. Okay, so that's definitely better. It's not perfect. It could still be a bit better, but in the interest of time, I'm not going to play around with all of the different parameters and all of the different settings and things that we could change. But if you had time, if you were trying to actually do your own pipeline, you could change each of these as many times as you want, individually, together, and just see what gives you the best outcome. So now after I've done that, I have got a filter object step in here, and I'm going to say filter out cells 1, remove any that are touching the border, and rename those objects cells. You can also say, would you like to relabel additional objects to come along and match the filtered objects? And yes, I absolutely do, because I want each cell to have a nucleus. So I'm going to say, yes, please select the additional object to relabel nuclei 1, relabel them as nuclei. And so now we're going to have cells 1, nuclei 1, which are all of the cells and nuclei, and then cells and nuclei, which are just the ones that are not touching the border. And that's important for the next things that we do, things like measuring the object's size and shape. So here we've got all of the different objects. You might call them stencils or masks, like these different object regions, definitions. So you can tick any of the boxes. I'm going to tick cells and nuclei. Certain key features, you probably want to say no, unless you have a reason to do that, which we can talk about another time. There's also some other advanced features. You might not want those right away, but I'll obviously have a look in the question mark if you're advanced feature curious. And the reason why, again, we've filtered out the ones that were touching the border is because we don't want to measure the area of a cell that's half cut off outside of the field of view. We can also now measure object intensity. So we can say, right, measure the cells, measure the nuclei, and I want to measure the actin, the DNA, and the EDU channel. I'm not going to measure the rescale actin intensity channel because we only use that for segmentation. Okay, so now that we've done those things, so we can step through. So there you can see we've removed anything that's touching the side. I've unticked the eyeball on the measurements. If I didn't, you'd get a pop-up that shows you all of the different things that you've measured. And again, you can always look at the module help to see what do all of these things mean. So at that point, we could be done. Maybe that's everything that we wanted to do, and we just want to export each individual cell with all of those things that we've measured, and we can skip straight to export to spreadsheet. But there are some interesting things that you can do to help you do some sort of quality controls and sanity checks. You should never only get your parameters optimized for a single image, because maybe you just happen to choose the one weird image that is not like the others, or maybe different treatment conditions cause different like cell shape changes or differences in intensity, and you want to find either a sweet spot where you get really good segmentation for all of those, or you might need to divide it up because you just can't find a set of parameters that works for everything. If you're doing some more complicated things where you're maybe calculating ratios, or you just want to see what kind of intensities you're getting or what kind of sizes or shapes you're getting, you can do some kind of interactive modules, which I tend to use when I'm making my pipeline. But then when it comes time to actually run the whole analysis, I will just untick that box, because I don't need, if I'm doing a batch analysis of 1,000 image sets, I don't need this display data on image to pop up. So what is this display data on image? Well, you can choose anything that you want to see, any of the things that you've measured, and you can overlay that on the image. So here we can say, yeah, pick the nuclei, and I'd like to see the intensity. Which of these intensities? The mean intensity in the EDU channel, and I would like to display that on the EDU image. And so that's going to give me a pop-up, which is mostly dark because most of these cells did not incorporate EDU. So you can see that the baseline of the mean intensity of the EDU negative cells is 0.02. But the cells which are EDU positive, we've got 0.29, 0.1, 0.12, 0.11. So that's quite helpful if I want to, say, make a threshold or a cutoff for what I consider an EDU positive versus an EDU negative cell. This kind of information is also helpful if you're looking to do like a background subtraction or a background correction, because you could define the foreground and the background if you've got a good amount of background, and find the mean intensity there, and then use that to do like a background subtraction. So this is quite helpful, but you could display it on anything. I could say, actually, let me show me the EDU intensities overlaid on the DNA image, or whatever you want to see there. You could also look at the integrated intensities, so that would be the sum of all the pixel intensities, say in the DNA image. And here we can see, so I remember that this was a cell that had incorporated EDU, so that means that this cell is in either S phase or G2. And so its total DNA intensity, its integrated intensity, is 508. And here were some EDU negative cells, which are 290, 300, 283. So these would be DNA content of 2N, and this is a DNA content of getting close to 4N. Here's another one that I remember was EDU positive, and this is 628, so that's about double the 300-ish that the other non-EDU positive, so cells in G0 or G1 DNA intensity. All right, so I'm just going to untick that because, I've seen it, I've played with it. I don't need to run it in the batch analysis. Here's another one, which is display histogram. And so here, let's look at the integrated intensity of the DNA. And we can see that most of our cells are in this region of about 300, but we've got a few cells which are our SG2 cells. And we've got some here which are lower, and I suspect that these are probably cells which have been either missegmented or like a dead cell that was not filtered out. Often if you're seeing cells that get cut in half, right, the integrated intensity, if you cut the nucleus in half, would be half of the total. So there's other things that you can look at here in the data tools. You can look at a scatter plot, you can look at a density plot, you can display anything you want on the images. Also in the data tools would be things like calculate math. So if you wanted to, say, take a ratio, maybe you've got phospho P53 antibody and a total P53 antibody, you could use a calculate math. I'll add it in so you can just see what it looks like, where you could take two measurements and divide them by one another. So you would choose, say, the object and the mean intensity in one channel, we'll just call this the DNA channel, versus the mean intensity in another channel, say the EDU channel. We could also do something with area shape. So we could say, give me the nucleus by cell area ratio. Importantly, in your output measurements, don't use spaces. CellProfiler does not like them. If you want to have a space, use an underscore. And you can mix and match. So I can use nucleus area divided by cell area, for example. And that will give me a ratio of the nucleus area to the cell area. Okay, so here I put a filter object step where I want to define a group of cells as EDU positive so that when I export, I could get the count of all the cells and the count of the EDU positive cells, and then I can just take that ratio or present that in my graph however I want to do it. I could look here on the display data on image or in the display histogram, and I could look at, say, the histogram of the EDU positive to see where I wanted to draw that line, and maybe I draw it at like 0.04. Untick that box just for the moment too, which is what I did, and maximum value is 1. There are other ways to calculate, say, percent GFP positive, percent EDU positive. I will put a link in there, but there's a really good YouTube channel by Beth Simony, who is one of the CellProfiler developers, and she's got a whole little tutorial video on all of the different ways that you could do this. This was like her least favorite way, so it's worth checking out if you want to know kind of more sophisticated ways or ways that might work in one context or be optimal in a different context. All right, so that's going to define a new subset of objects. So if we click through, now we can just check and see how that went. And yep, sure enough, here are all of our nuclei, and here are the EDU positive ones. Okay, so finally, we're going to, once we're happy with all of that, we're going to export the data. So I'm going to leave the test mode. If I was going to run analyze images and start the batch analysis, I might want to untick these boxes. Actually, these ones could be, these little eyes could be left open because we're not actually running these. If you leave the little eyes open, it's going to give you a pop-up window, but with every image set, which might make it take a little bit longer, or you might not want to see that because you're doing something else. So we've got a little X here. If you hover over, it can give you some information about why it's not happy. So here, pretty obviously, we haven't actually told it where to save the data. So you can save it in the default input folder, default output folder. I usually click elsewhere because I don't want to save it in this the analysis CSV files in the same folder as my images came from. So I can say, put it in Documents, CellProfiler. Add a prefix is really helpful. So I can say the prefix is demo 2025, maybe nuke and cells, and the underscore will just make it easier to read. Add image metadata columns to your object data file. I would definitely click yes here because that is in your output spreadsheet where each row is an individual nucleus or cell or object. You want to know where it came from. So yes, we do want to do that. You can also say, do you want the image file name or do you want the folder name? Those could be helpful because maybe you've got, maybe you're doing a batch analysis and you've got multiple different folders. If you don't select the measurements to export, if you say no here, it's going to export every single thing that you measured. Now, on these measurements, so on object intensity, no, sorry, I didn't mean to add it there. I just wanted to look at the module help. It's measuring all sorts of things. Maybe I want those in my output, but maybe I only want some of them. So if I don't want to export everything, I can say select the measurements to export, and now I can choose what I want to export. But you have to actually choose all of the things that you want. So first things first, I definitely want the count, and I want, say, the count of cells and the count of EDU positive cells. You could also get maybe the first cells before the object filter if you want to know like how many it removed, compared to how many it found initially. And I want to export some metadata, so I want the column, the position, the row. I'll take the scene and the well also. Channel is an option here. Don't tick channel, because each data set has, or each set of files has 3 channels. So like this will not come out. You won't get anything in that. There's other things you can also choose here, but those are the important ones. And now for cells, maybe I really just want the area. Maybe I want like the form factor. Maybe I want the radius. For intensity, maybe I want the active intensity. Probably doesn't have any DNA or EDU. But maybe I'll take the mean intensity of all of them, because if there is some high value in the cell DNA, that might flag that there's some sort of problem with the segmentation. You can also put the location, so you can say the XY coordinates, and you can ask for the math that you've done. And again, for the EDU positive, you can tick all or none of these boxes. The parent would be the number of the nucleus that it came from. We haven't actually made any measurements on it, so maybe we just want to get its location. And again, for nuclei, we can say we want some of the information, maybe the solidity. Children would be, do you want to know what EDU positive number goes with that particular nucleus? And then here, maybe I want the DNA and the EDU, and maybe I'll take the mean intensity for everything. So if you just, anytime there's a little plus, you can click on that and see what all the options are. You can say none, all, or some. Tick the math there too. So once you're happy with that, you can then decide, would you like to calculate the per image mean, median, and standard deviation? So if you say no, you'll just get spreadsheets where every row is a single object, a single cell, a single nucleus. But here I might want to say, yeah, give me like the mean per image and the standard deviation per image. This can be really helpful for quality control. It's also very useful because this is also going to give me the count, because I've chosen to export the counts here. And so I might want to see, actually, I want to know in every image what was the count of cells and what was the count of EDU positive cells. So now if I have chosen this, I want to say, okay, the data that I want to export. So I would like to export the image information, so the mean and median as well, and standard deviation per image. Use the object name for the file name. Yeah, sure. And then now it's going to ask, okay, what individual objects do you want to export? So I would like to get the nuclei. I didn't really make any measurements on the EDU positive, but I made measurements on the cells. So what I can say is, I can either export all of the information about the nuclei into one CSV file and all of the information about the cells into a second CSV file, or I can say, actually, give me one CSV file. Each row is a cell that has measurements about the cell body and measurements about the nucleus. And so I could say yes there. And now I'm going to get two sets of things. I'm going to get the image means, medians, and counts, and standard deviations and counts. And then I'm going to get another spreadsheet which has all of the individual cells with the measurements that I've chosen about the nuclei and the cells. You might want to save your project again. And now I can run this by clicking Analyze Images, and then it should give me, after a little while, the spreadsheets that I'm after. So my advice to you would be not to start with all, 1,000 of your data, but to import, just to bring in a few from column A, a few from column B, try some things out, and then go all the way through the Analyze Images stage. Look at your CSV files that you get out and make sure that you've got all of the information that you want, that you haven't skipped ticking any boxes. Or if you've got some numbers that look weird, make sure that in your calculate math, for example, that you've actually got the right measurements. And I would do that on a short little subset of the data, and then go back and do your whole long batch analysis that might take some time. Also, when you're in test mode, you have the option of skipping to the next image set. So if I click that button, it's going to go from scene 638 to scene 806. And so hopefully when it shows me the primary objects, it's going to look different than the picture that we saw before. Hiding from me. Oh, you know what? I didn't tick the eyeball, but let's say for the sake of argument that it did. And so you can just keep skipping ahead to the next image set. If I go next image set again, it's just going to take me back to the beginning because I've only got 2 image sets here. Okay, so I think that's just about out of time. I think I've run a little over time. But before we go, there's one other module that I would like to show, and that is tertiary objects. So got primary objects, blobs. Secondary objects, build out from the blob. Tertiary objects is going to relate different blobs to one another. So the most obvious tertiary object is cytoplasm. So the larger identified object are the cells, the smaller identified objects are the nuclei. So the cytoplasm is going to subtract the nuclei from the cell. You can say, do you want to shrink it? Yes or no, just to make sure that you've really got something in there. And so you could do this with cells and nuclei. You could also do this with, like if you had cells and you had vesicles, you could identify the tertiary objects as being vesicles inside the cell or inside the cytoplasm. Okay, so with that, I will end. I'm going to stop recording.